So whenever you're ready, Jonathan, you can start.
Okay. Good afternoon for everyone. Today I'm going to present the paper, a comparison of automatic cell identification methods for single cell RNA sequencing data. This paper was published on Genome Biology Journal. This paper was published on 2019. Just for introduce in the context of the paper, in the pipeline of bioinformatic analysis of single-cell RNA-seq, we need is first to alignment, after quantification, normalization, and after that we do the dimensionally reduction and clustering. And once we have the clustering, we need to identify the cell types. Usually, this annotation of the cluster is manually, but the manually annotation have some challenges and difficulties because sometimes you need to spend much time trying to find out what kind of the cell type is. Frequently, this annotation is not based on standardized ontologies of the labels, and frequently these annotations are not reproducible across different experiments. So the paper said that for overcoming these challenges, there are some automatic cell identification methods. These identification methods are based on correlation analysis, or sometimes are based on marker genes database that they use for the annotation, or there are some tools that use supervised classification methods. The principal objective of the main objective of this paper was to compare methods to assign cell type labels to clusters, and they performed benchmarking automatic cell identification. They evaluate 22 classification methods and analyze 27 publicly available single-cell RNA sequencing datasets. And the evaluation classification was, they used two experimental setups. The first was an intra dataset setup. They use a 5-fold cross-validation within each dataset. And the second was an inter-dataset evaluation across datasets comparison. The classification methods that they used were divided into categories. One category was the prior knowledge methods, for which either a microgene file is required for an input or a pre-trained classifier for a specific cell population. And the other was supervised methods, in which is required a training data set, a label with the corresponding cell population in order to train the classifier. The classification methods use a language like R or Python. And there are several types of methodologies that they use. Okay, the dataset that they use are from different tissues. For example, pancreas, lung cancer, musculus. They use also visual cortex, PBNC, and there are also some variation in the protocols of the dataset. They select a dataset that, assim, não sei, que eles são muito variáveis. Eles precisavam que fosse muito variável. They needed that the dataset were variable in protocols and tissues, the number of genes, and the number of cells, and the population also. So the first evaluation was the intra dataset. So they divide a dataset in 5 groups. They try to each group conserve the same proportion of the cell types, and they use the original annotation for the train data set. For the train, they found that, for example, in pancreas, this is a lung cancer and musculus. The CBM reaction method and CCS spread method was they obtained the best scores for the performance. The score that they used for the evaluation of the performance was the if1 score. This F score is a measure that is accuracy and is calculated for the precision and the recall. So values closer to 1 indicate that the performance is bigger, and close to 0 is lower the performance. So in the first intra dataset evaluation, the best performance was for SBM. But when they analyzed a PBNC datasets, they found that the performance of the majority of the methods was lower in comparison with other tissues like pancreas or T cell lung cancer. Sorry.
Jonathan, there are some cells. They are gray in the table. Do you know why? I mean, for example, in PBMC, the second row, there's a gray cell there, and
Here?
it's in the top, the second row from top to bottom. Sorry, like in the PBMC, the last column. Do you know why they have this? I mean, it's not a 0. It's just gray.
The gray box.
Yeah.
They said that they can't perform the evaluation because the proportion of the cells are not equal.
Oh, okay.
Because the Shang datasets, they divided into groups. The first was sorted, and the second was a dataset that have a deep annotation. So because this dataset is so deeply annotated, the proportion of the cells are not equally, so they didn't perform the.
Oh yeah, all right. Thanks.
They evaluate also the percentage of the unlabeled cells. So they found for example, in PBNC, there are 23.5% of cluster that was not annotated. For a deeper annotated dataset like the shank that use greater a greater number of cells, they got a greater number of cells that were not labeled.
Jonathan, the order of the names of these programs are from the first one is the best and the second is the second best, so on?
Yes, they order by performance. The first was the best results, and the last one was the worst results.
So until now, the best ones are SVM rejection and SCPRED, isn't it?
Yes.
And both are from R. I know that SCPRED is from R. Do you know SVM?
SVM. Python.
Python. Okay. And SCPRED is from R, isn't it? No. SCPRED. Sorry.
SVM. SVM, as I can see, is a classification machine learning algorithm in Python. It supports vector machine. and they show that they use a linear kernel for the classification. It's
Okay.
so it's probably the scikit-learn.
Okay, thanks.
Okay. So for the category of methods that use prior knowledge, they evaluate if the number of number of markers change the performance of the classifier. So they observed the number of markers didn't have a significant correlation with the F1 score, but they observed that the mean of the marker expression of the marker genes, they are positive correlated with the performance, because when they observe that the marker genes have a mean expression higher, they obtain a higher F1 score. Similarly, they observed that technical factors like dropout rate are correlated significantly in that evaluation. They observed that the dropout rate are correlated, they have negative correlation. So the technical factors influence the gene annotations and the classifiers. They use a value beta. The paper didn't explain what is this value beta, but I searched in another paper, and I found that is a value that is calculated for the proportion and the expression of the marker. I don't understand really how this calculus is made, but they said that this score is, for example, calculated based on the mean expression of a marker, and if you have a beta score high, means that you have a probability that this marker is more specific for this cluster of this type of cells. So when they evaluate the performance of this method, they observe that this value is positive correlated with the performance of the classifiers. They also analyze the performance and the complexity of the data sets.
Can you go back to the beta the equation?
Here.
Markers. What is this? Plus a small constant to avoid division by 0. Go to 1. Okay. So just, F1 score, just go back one, just to keep it clear. F1 score is a metric to evaluate how good the classification is. So the higher the F1 score, the better is the classification, right? F1 is a harmonic mean of the classification metrics. And I'm just trying to understand what is the beta they use, but it's probably one of the hyperparameters for the classification, right? I think, but anyways, go ahead. Thanks.
They evaluate also the complexity and the performance, and they observe the more complex data sets have lower F1 score. They want to know if the performance is affected by the protocols, because in this analysis they use the same sample that was processed in the same lab, but they change the protocol that used to analyze the samples. So they use a training set for this protocol, and this is the test, the test. So they found that in this evaluation, the protocol, the change of the protocol doesn't influence much in the score for the performance. Meu Deus. Desculpa, sorry. Sim, eles utilizaram a mesma amostra que foi processada no mesmo laboratório e eles queriam saber se realmente aquele mudança do protocolo influencia no performance do classificador, e eles observaram que praticamente tem o mesmo rendimento. Então, eles continuaram, porque this dataset is very simple. There are three types of cells. So they use this dataset that is from PBNC, that is more complex because there are more types of cells. So they apply the same strategy. They use the same sample, but use different protocols, and they compare each protocol for, o sea, eles compararam cada protocolo com, o sea, treinaram com um protocolo e compararam com os outros, e eles foram fazendo as comparações para cada protocolo. Então, eles, nesse caso, in this case, they observed that, for example, this CINA method was correlated with the best performance, and the single cell net was also, también obteve un bom performance. Então, aqui, a linha vermelha indica que este daqui foi o dataset de treinamento e esse daqui os de teste para cada um dos protocolos. E o resultado foi esse, o CINA e o Single Cell Net foram de melhor performance. They analyzed also the percentage of unlabeled cells, and they found that also CINA and Single Cell Net was the best classifiers for this data set. They separate the two methods that use prior knowledge, and found the method. They found that method CINA was the best, get the best performance. They use the same strategy. In this case, they use different protocols and different samples, because they want to know if the performance is, o sea, eles querían saber si o rendimento desse classificador era igual em diferentes protocolos e em diferentes tipos de amostras. Então, eles utilizaram outros tipos de amostras com diferentes protocolos e eles ainda encontraram que o performance do classificador era bom. E eles evaluaram também a porcentagem de células que não foram anotadas. E aí, apesar de que, por exemplo, em alguns protocolos o porcentagem era muito alto, como 60% neste caso, as porcentagens em outros protocolos eram bastante baixas, 2%, 10%, 7%. Então, um classificador bom. They try to analyze this performance between two types of tissue. They compare the mouse and the human of the cortex, and try to evaluate if the classifier, se o classificador tem um performance bom quando comparados diferentes tipos de tecidos, e eles encontraram que o SVM, ele era o classificador com melhor performance, tanto na pontuação de performance de F1, e também obteve baixa porcentagem de células não anotadas.
Jonathan, uma pergunta. Quando ele fala aí de dataset de treinamento e de validação, tá, eles usaram um dataset diferente para treinar e um outro para testar.
Sim, eles queriam saber se, digamos, se eu utilizo um dataset de treinamento de rato, ele era capaz de identificar as células, por exemplo, de humano. O sea, eles queriam saber como ele ia funcionar se eu treinava com um dataset de rato e eu testava com humano. Então, eles foram trocando. Então, eles primeiro utilizaram em um grupo, eles treinaram com rato e depois treinaram com humano. E este outro também é de humano, de córtex. Então, eles treinaram com mouse e testaram com humano. E foram testando os três. Então, todas as combinações possíveis, eles testaram.
Aí o que eu fico encucado sempre nisso, tipo, nesses estudos, é que você usa um tecido para ver o tipo celular de outro animal. Como é que você sabe que o tipo celular é o mesmo numa espécie e na outra, saca? Tipo,
Eu acho que o objetivo
Thiago,
dele era saber se ele ia classificar de maneira errada, talvez porque se
Thiago,
você classifica como, digamos, você tem um neurônio de rato e um neurônio humano, eles são diferentes. Então, se você
mas eles são
treinou
fundamentalmente diferentes, assim,
sim,
eles são espécies diferentes, né?
sim.
Tipo, então é muito complicado você dizer que os dois são a mesma coisa entre espécies só a partir da transcriptômica. Sei lá,
Thiago,
acho que tem algumas dificuldades fundamentais aí na abordagem deles. Assim.
geralmente, Thiago, os genes marcadores de tipos celulares, eles são bem similares entre espécies. Tipo, uma célula B o marcador de célula B, por exemplo, de PBMC, né, no humano, geralmente são quase os mesmos ou são bem próximos, sabe, pelo menos assim, que eu tô vendo que o pessoal me fala, tipo, quais são esses marcadores aqui em camundongo? Quais são esses marcadores aqui em humano? Geralmente é a mesma coisa. Até nesses sites aí de anotação, sei lá. Eu sei que não é a mesma coisa, mas...
O PangLauDB, que eles até usam nesse estudo aí, ele mistura o marcador de camundongo e ser humano. Só que isso é uma bagunça tremenda. Não existe identificador comum de gene para camundongo e ser humano. A gente usa ou de symbol de humano ou de symbol de camundongo. A gente não tem um identificador comum padronizado, sabe?
É o gene ortólogo.
É,
Por exemplo, sei lá, IL-1 beta. A IL-1 beta no ser humano é uma. Aí, às vezes, sei lá, no ser humano tem IL-1 alfa e IL-1 beta. Aí, às vezes, no camundongo só tem IL-1, né? Não tem essa subdivisão de IL-1 alfa e IL-1 beta. E, às vezes, você usa essa IL-1 para identificar alguma coisa.
claro, mas não existe identificador único. Isso aí é
Não,
tudo.
sim.
Não existe uma identificação formal oficial para um gene multiespécie. O que o pessoal faz é que transforma o gene do camundongo em maiúscula e assume que é a mesma coisa. E aí, tudo bem. Não, eu não estou dizendo que, tipo, tem mérito aí o estudo, Jonathan, eu não estou criticando, estou falando que eu fico muito pegando, assim, do que, tipo, você tem, tipo, parece que ele está falando, não, todos os tipos celulares que existem em camundongo existem em humano, né? Quando você faz um estudo desse. E isso não pode ser verdade. Isso não pode ser verdade.
É o que falta, na verdade, é um referencial, né, para você poder. Mas eu acho que eu faria o que o Edson falou, pega o ortólogo.
Por exemplo, macrófago, né? Macrófago em humano tem.
Mas é porco. É tipo, é porco, né? Não é uma ciência fina, assim, tipo, você está vendo a biologia da coisa. Você está meio que fazendo vários pulos hipotéticos de que as coisas são iguais. Assim.
Jonathan,
Sí, estaba pensando que eso depende también de qué tan profundo es la anotación, porque tal vez si uno escala, algunos grupos celulares son bastante semejantes, pero si uno va más hacia los subtipos, ahí realmente existen bastantes diferencias. Y es lo que muestran aquí, porque por ejemplo aquí están analizando solamente 3 tipos de poblaciones y aquí están analizando 34 tipos de células. Aquí sí los porcentajes de performance son muy bajos. que para mí realmente tiene sentido, porque realmente son dos especies totalmente distintas. Aquí, por ejemplo, el performance es 0.18, es un performance muy bajo.
Jonathan, você sabe dizer se a anotação que eles fizeram foi essa anotação mais geral? Por exemplo, macrófago só chegou a macrófago. Ou macrófago, se ele foi subdividir o subtipo de macrófago. Da mesma forma, tipo, célula T. Foi só a anotação célula T, ou não? Foi TCD8, TCD4, Treg e assim por diante.
Bueno, aquí no, porque estos son de córtex. Es aquí, ahí, PMC, este de aquí. Então, por exemplo, aqui, este dataset daqui, ele se foi anotado dessa maneira. Então, por exemplo, eles... es que eu teria que mostrar o artigo, melhor, vou mostrar o artigo aqui. Especificamente, os tipos que eles utilizaram. Aqui. Então, este daqui é PBMC. Então, eles... Então, meu Deus, Espanha, meter. Então, eles... aqui, eles classificaram CD8 citotóxico, CD8 naive, CD4 T-Rex, memória, naive CD4, T-Helper. E aí, eu vou mostrar mais na frente que o rendimento cai muito, porque eles analisam, por exemplo, aqui os true labels, que são as anotações próprias do artigo, e as que foram preditas pelo classificador. Então, por exemplo, aqui, subgrupos mais específicos de células CD4, eles estão mais correlacionadas. E lá no performance foi bastante baixo, caiu muito quando eles querem fazer uma anotação mais profunda. O performance do classificador é bom quando você quer, por exemplo, identificar CD4, mas você não sabe se esse CD4 é naive, se é regulador, se é naive, se é memória. Aí não tem problema, mas quando você quer saber se esse CD4 é regulatório, é naive, tem outra coisa que você quer saber mais aprofundar sobre esse subtipo, aí o performance cai. Que é o que eles... que é aqui.
Beleza.
Então, aqui... Então, aqui é o mesmo análise, só que eles estão mostrando a quantidade de células que não foram anotadas. Aqui a porcentagem é muito baixa. Para mim é um mau resultado, porque se você está comparando rato com humano, em teoria, tendria que ter uma porcentagem de células não classificadas muito alta. E você vê que não, a maioria são 0, 0, 0. Então, digamos que esses classificadores não são muito efetivos, porque se você quer saber determinado grupo de células, mas você de repente achou um novo tipo de célula, o classificador deveria ter essa capacidade de dizer, não, este tipo de célula aqui não encaixa em nenhum. Então, ele não identificou nenhum cluster assim. Eles tentaram fazer outra abordagem também. Eles... eu não sei como é que funciona isso, mas eles alinharam os datasets de diferentes... Ou seja, 4 datasets. Eles utilizaram 4 datasets e eles observaram que existia como um efeito de grupo, um efeito de lote. Então, eles alinharam esses 4 para fazer este teste aqui. Então, eles compararam os datasets que não foram alinhados, ou seja, com o efeito de lote, e depois corrigiu o efeito de lote, para saber se o classificador ele mudava sua performance. Aí eles observaram, por exemplo, que quando você não corrige os efeitos de lotes, os classificadores CSBL e SVM, eles têm uma melhor performance. Mas quando você corrige o efeito de lotes, aqueles métodos que têm melhor performance são KNN e SVM, rejection. Então, foi o que eles analisaram assim. Aí fica uma dúvida, porque aqui, por exemplo, o efeito não foi corrigido, mas você tem um bom performance. Possivelmente você está notando de maneira errada as células, eu acho. Não sei o pensamento que eu tenho sobre isso. Né? E eles fizeram também uma análise de controle negativo. Então, alguns classificadores trazem uma opção que chama de Rejection Option Evaluation. É quando eles identificam que um grupo de células eles não pertencem a nenhum dos outros que já estão anotados, então eles classificam como um tipo de célula novo. Então, o que eles fizeram era assim, eles utilizaram dois datasets que são totalmente diferentes, que não têm absolutamente nada a ver. Então, eles, por exemplo, como dataset de treinamento, utilizaram o PBMC. E para teste utilizaram câncer de pulmão. Então, em teoria, a maior parte ou a maior porcentagem de células anotadas deveriam estar como não classificadas, um label. Então, eles observaram que os classificadores ainda têm uma performance muito baixa para esse tipo de função. Então, eles não conseguem, digamos que de alguma forma, identificar esses grupos de células que não têm nada a ver com o que está sendo treinado. Eles também avaliaram algumas outras características, como por exemplo, que tanto influencia o número de features, número de genes no performance. Então, por exemplo, eles observaram que o SVM era um dos classificadores com melhor performance e que não está influenciado por o número de genes. Eles observaram que, por exemplo, a quantidade de clusters que não foram anotados, eles aumentam na medida que você aumenta o número de genes. Então, segundo eles, é importante realizar uma boa seleção dos genes que vão ser utilizados para a anotação, porque na medida que você vai aumentando o número de genes, você vai diminuindo em alguns o performance, e em muitos deles você vai achando maior número de células que não são anotadas. Também analisaram performance mudando o número de células e observaram que o número de células não influencia muito o performance nos diferentes classificadores. Tem alguns nos quais se influenciou bastante, por exemplo, o LDA, ele cai muito quando você aumenta o número de células, mas ele, por algum motivo, ele depois, quando você sobrepassa um determinado número de células, ele melhora o performance. Também eles analisaram o tempo de corrida dos classificadores. E nessa prova, o melhor foi, que foi esse daqui, porque as cores são muito semelhantes, o NMS. Mas, em geral, são classificadores que empregam bastante tempo. São 10.000 segundos. 10.000 segundos são mais ou menos 37 horas, é bastante tempo. Então, aqui utilizaram o tempo segundo aqui como modifica o tempo segundo o número de células. Então, a maior número de células tem maior tempo de corrida. O nível de anotação, ele não influencia muito no tempo de corrida. E ali no final ele faz como um resumo das avaliações que eles fizeram. Então, por exemplo, eles classificam como bom, intermédio e baixo o performance para cada um dos classificadores e eles ordenaram segundo as calificações e eles observaram que, por exemplo, o SVM ele teve o melhor rendimento tanto nas avaliações intra-dataset, inter-dataset, nas provas de rejeição e no tempo de corrida. Pronto. Então, eles finalmente concluem que a incorporação de marcadores não melhora o performance dos classificadores, especialmente em PBMC, porque o que realmente melhora o performance é a especificidade dos marcadores, como foi mostrado na gráfica que está o value beta, o valor beta, que está correlacionado positivamente. E também diz que o performance entre os métodos cambia en dependencia de los genes que se están utilizando. Entonces, é importante realizar uma boa seleção dos genes que vão ser utilizados para anotação. E eles recomendam que, para uso geral, pode ser utilizado o SVM como classificador por os resultados que eles obtiveram. Também eles compartilharam como o workflow com os códigos, por se alguma outra pessoa quer reproduzir o trabalho ou quer, por exemplo, testar com outros datasets.
Jonathan, do you know if the authors of these papers have something related to this SVM rejection method?
No.
Because
No.
sometimes they are just selling the program.
So SVM is really it's decades old. It's really the standard machine learning algorithm for pretty much any kind of classification. It's kind of the baseline that people use. I mean, it's SVM and random forest.
Mm-hmm.
And that's one point that's for me. It's they definitely didn't invent SVM. That's definitely not their invention. But I have never seen anyone actually use in a paper. So we used SVM to classify cells. I haven't seen this ever.
Neither me.
Not even once. So I mean, why is it? Because it's a bad recommendation. It's not a good recommendation. And why is that so? I mean, I've read this paper before, and it has a lot of numbers and it does a lot of things, but maybe Jonathan, for you, like, what were they trying to do? What's the answer that they were looking for? Because if they were looking for the best, that doesn't exist. I mean, it's best for something. For me, it doesn't look like they have a use case in mind. They just want to find a best or whatever that means. I mean, I don't know.
Yes,
Mauro, acho que você está mutado.
Sorry, go ahead, Jonathan. I'll make my point later.
Yes, I think that the author tried to find the best classifier, but I think that this is a bad conclusion, because when I think that the classifier, I don't know how to say that, but yes, you have a good score, but that doesn't mean that the annotation is good, because eu vou falar português. Eles querem achar o melhor classificador. E eles botam aí que você tem um score bom, que você tem uma baixa classificação de células não anotadas, mas no final você vê que eles não conseguem fazer uma diferenciação real dos diferentes tipos de células. Então, no final, eu acho que ainda você tem que fazer de maneira manual. Então, assim, para mim, a conclusão é isso. Assim, eles fizeram muitas análises, tudo bem, tem um score muito alto, mas no final, quando você olha as tabelas de correlações, que eles colocam também no artigo, você que esses subtipos que você precisa achar, eles não conseguem identificar. Então, não faz muito sentido utilizar esse tipo de classificador. E aí, no final, se você tem que utilizar manual, e se você tem que utilizar, por exemplo, alguns marcadores, e saber selecionar esses marcadores, então, ainda não, eu acho que ainda não é tempo de utilizar esses classificadores. Eles precisam de mais tempo para serem desenvolvidos. É isso.
Mauro, Ícaro, pode falar.
So that was exactly the same thing I was going to mention. Like, they tested several classifiers in different data sets, and they got different results, like, and tried to understand which is the best classifier. And of course, it depends on several parameters and hyperparameters that they put it on it. And in conclusion, you have to do it manually. That's basically what I thought.
Yeah, I agree with Jonathan and Mauro. I think in this paper we see the problem that we face when we try to annotate using these different methods and approaches, complex data sets like PBMC, for example, or try to have a deep annotation level. And I think this paper just confirms what we saw before. Like, we cannot guarantee a good quality annotation when we try to use these automatic tools. So we need to go and do it manually. But I think the most contribution of these papers provide for us is like if we are dealing, for example, like right now in COVID Atlas, that we have a lot of data sets that the authors didn't provide annotation. So we can use, for example, one of these methods just to do primary annotation, just to see what are the T cells, what are the clusters of, for example, macrophages, and this just to know the populations of cells, the main populations of cells, and based on that, do manually a manual annotation on these specific clusters to do this annotation process. So I think, at least for me, this is the most contribution that we have when we use these tools, like just to see what are the T cell cluster and what are the other clusters that we can annotate, and based on that, go deeply and try to annotate manually. This is typically what I do when I try to annotate in single cell data sets.
I wanted to say one thing, that while doing with COVID Atlas, there was one paper which just provides the annotation. The authors provided the annotation, but somehow I was checking the other three methods. I think single R, azimuth, and one more, as catch. So I did annotations using both three programs, and I wanted to compare how does it integrate something somehow with authors one, but I didn't find any of the annotation program matches with the authors one. I don't know then which one is correct. Does I believe authors or what?
It's a mess. I mean, because in this case, even the author's annotation, there's nothing that says what a neuron is anywhere.
Yeah.
It's a matter of like you have an expert, he or she,
Yeah.
they're looking at the data and they say, okay, these groups are new. But if you look at PBMC data,
Mm-hmm.
CD4 cells that are labeled as CD4 T cells by authors
Mm-hmm.
often are CD8 positive and CD4 negative. And you also have the opposite. So, I mean, because it's in the middle of the cluster, so
Of course, yes. They are close. Yeah.
But if you take one cell like this and show it to immunologists, say, okay, look at this cell. It doesn't express CD4 and express CD8. It's not a CD4 T cell, but it is. And so it's. I think that the point that I'm trying to make is that it's a mess. So we don't look for like achieving the best annotation ever, because we won't be able to do that. So that's in this Elder is very very wise when he says that we have to use the annotations by the original authors, because then we don't have to
mug our mind there. Yes.
because in the end it's like an art. You look and you have the expert and call it, and that's hard. That's hard. So yeah.
I wanted to ask one question to the whole group, that the papers which doesn't provide the annotation, because if authors provide annotation, I somehow try to see that if I can find the annotation from authors. But if in case which I doesn't find any, what I do, like I use tools or I use manual. Of course, manual annotation takes time, but both.
Well, I think it depends on the data set too. But one thing that we depend when we are trying to annotate the data set is the clusters. So depending on the metrics that we use it and the resolution that we got from the clusters, maybe this will affect the annotation part too. So it's you always need to check if the clusters that you're using or the resolution it fits to the data set that you're looking at. So I think this is a problem too. Like, how can we select like the resolution that we want for the clusters? And
So I was trying until now. I was trying to look for the resolution which authors provided sometimes, but the cell types they provided in the paper, the dim plot. So I used to combine them sometimes, but is that right way? I don't know. I was thinking like this paper does provide some recommendations, but I guess it's not.
When you say resolution, is that the number of clusters?
Is a parameter of when you are producing the cell clusters that you have to put like between 0 and 2 around this. And if you put a big resolution like 2, you will find a big number of clusters, like 30, 40, something like this.
Like
And if you put a small resolution like 0.4, 0.2, you're going to find like 10 clusters, 5 clusters, something like this.
And what is the most used number of resolution?
It doesn't
exist.
It depends on
on how much you wanted
the
separate.
paper, but sometimes like the paper uses just 0.4, and I can put 1. I'm gonna find more clusters, but in the end, I'm gonna find the same clusters. This is the point. So if you're trying to find subtypes of this cluster, like the author just provided the T cell annotation, and I want to find the TCD4 and TCD8. So I can try to put a bigger resolution to try to specify the subsets of these clusters, and I will do the manual annotation. Oh, this subset of these two are T cells, but this one is CD4 and this one is CD8. But in the end, in general, when I'm doing this annotation, I use a little bigger resolution just to find more clusters, just to guarantee that I have more clusters than the authors. And in the end, I have at least the same clusters, at least. But in my opinion, I don't know if you noted this, Icaro. I think you analyze at least three single cells of PBMC. And did you note that the same, usually we have the same gene markers for the same cell type in general? Like these cells usually have this one, and this one, and they are the same in the three data sets, for example. Did you note this?
Yes. This is something that I found. Like, for example, for neutrophils, for example, I found the same gene markers in the 3 data sets that I've compared. But all of the 3 data sets are from COVID, so I think this is okay. Like, it's comparable.
I was suggesting,
if you find different markers, then it's a different cell type. I mean, that's circular.
Yeah. So
but
you would expect to find the same cell markers.
But in general, I think that we have at least like the main markers for each cell type, at least for the general cell type. And I was suggest for Yuvica, like Yuvica, I think you can like create your table of marker genes using 3 or 5 data sets for the same cell type. Like for example, for PBMC, in general, for these 3 or 5 data sets, I have these genes for neutrophils, these genes for B cells, and something like this. I don't know if you have a better idea or another suggestion for we find out a solution for this.
Yeah, that was
I was
the
planning
idea
to
that we
do
had.
this for.
Yeah.
Yes, until now we planned like this, but I'm planning to use this strategy to those data set which doesn't provide annotation. In some cases where I could not find a way to get the annotation from authors, then only I will try to do this. I'm planning like this on the, yeah.
Do you guys have another possible solution for this problem?
No.
Thiago,
before the suggestion that you've said, we thought like another day we were discussing if it was possible to get all the annotations that the authors gave us, and of course separate by tissue or whatever type of cells, and then we can get like which are the main genes that are in common in all the authors' annotations. Then we would have like a more robust marker for a type of cell. But then we would need to reanalyze. Now that you said about resolution, we would need to set resolution the same for all the studies and redo it, because we have to have the same number of clusters, right, in order to. No, we don't. Okay, so yeah, that was one idea. Get all the annotations of all the authors, and also all the annotations from all the packages, and join this in one big table and try to see if there is something in common, and make like one type of common annotation somehow, if it's even possible, you know. But that would be, like Yuvica said, that would take so much time. Actually, this is another separate project that would take a lot of time to do. But I think that's the best suggestion that you gave to us. Just for those studies that don't have the annotation, we do this that you said. But I'm always gonna have like a flea behind my ear that like the author's annotations we cannot trust 100%, because how do we know if they're doing their work well, right? We don't.
It's a mess. I think that the best thing about this paper that Jonathan has presented to us is that it basically shows that it doesn't matter. It doesn't matter what tool you use. So my conclusion is very different from the authors, is that just use whatever it's more comfortable and then manually do the mappings and stuff. Because as Thiago was saying, even the original authors, we cannot be sure exactly, because there are no definitions of what the cell types are, and people will call same things by different names and different things by the same names. And yeah, so use whatever it is. Use one thing that is you're more comfortable with, and no one can say that, oh no, you're not using the best method, because there is no best method. Yeah.
Okay.
Yeah. I would be more comfortable if we could do this, like get all the annotations of all the authors, see if there's like a common thing, but that would be too much time to do. But I don't know. Complicated. Also, Jonathan found another paper, right? Do you want to mention this, or I guess we're out of time for today, but maybe we can present this other paper some other time.
Yes, but I want to ask for a recommendation, because I thinking here in a biological problem. For example, when we have a conventional dendritic cells, we have 5 markers that indicate maturity of these cells. For example, CD40, 80, 86, HLDR, HLDG. And the level of expression of these markers indicate the state of maturity of the dendritic cells. And this is biologically important, because if you have a state that is immaturity of these cells, maybe you have a low priming of these cells, and maybe you have a low adaptive immune response and bigger innate immune response, and this can produce an inflammatory response that is bigger than normal. And what would you recommend me is for find this in single cell? Because I would like to find a way to find this type of maturity of on the dendritic cell of another T cell, for example, CD4 or CD8. I think that biological is very important, but I don't know how to deal with this when I do the annotation, because sometimes the cluster is very weird, because usually you have two markers that is typical, but you don't found in this. And sometimes you have that it is expressed, but I don't know how to know if the expression is higher or is middle or is low, because if it's low, middle, or high, that indicates the state of maturation of this type of cells. I don't know if I explained well my question.
I didn't understand the details. So you want to see the state of maturation of dendritic cells in a data set, and you, I don't know. You can try in Portuguese, perhaps, Jonathan.
Sim, a questão é que eu coloquei o exemplo das células dendríticas. Então, digamos, temos 3 marcadores. Esses 3 marcadores indicam estado de maturidade. Analisar biologicamente o estado de maturidade das células dendríticas, por colocar um exemplo, é importante. Porque se você tem células imaturas, você pode ter uma resposta imune inata maior, e isso causa uma resposta inflamatória sistêmica maior. E no final isso pode levar ao óbito dos pacientes. Então, seria interessante, por exemplo, analisar isso. Mas o estado de maturidade não é a presença ou ausência do marcador, é o nível de expressão. Porque quando você lê os artigos, você lhe fala assim, não, a expressão é low, mid and high. Isso é possível fazer esse análise com... Ou como poderia fazer? Porque realmente não sei fazer isso. Qual seria uma recomendação para analisar isso?
Jonathan, I guess my recommendation is that many of those things, they come from proteomic, like from protein level experiments, so flow cytometry and stuff, and that doesn't always correlate with RNA. So I think that's the first thing to keep in mind. And the second one is that, I guess, you would have to get the data set and really work on it a lot of time. I mean, I don't think there's by now like a way to do this kind of things automatically. Well, I can say, oh, try do pseudo time or whatever. But the truth is, if you want to really find these differences, you have to familiarize yourself with the data set, so understand really what the distribution of the genes are for each different gene, which cells, and then this is going to eventually lead to the analysis that you want. So I don't think there's a easy way on that. So if you want to do things that are complicated, you will have to spend some time becoming friends with the data set and knowing the dirty secrets, and then you might find the different populations of the dendritic cells or not. But there's, I don't think there's a easy way to do it, right? I mean, but that's my 5 cents.
I might be wrong, but if you can find like the group or the cluster that have the cells that you want, let's say it's cluster 1 has all your dendritic cells, right? So you know those 1,000 cells are dendritic cells. So you can then subset the counts, which the genes that you want to see the expression, right? So you say, let's say there's 10 genes, I don't know, and you subset the 10 genes for all these 1,000 cells. After you normalize the counts and everything, you can just divide the expression by histogram. So you know, like, okay, these 100 cells have a low expression of the genes. These other 1,000, or I don't know, 500 cells have medium, and these 300 have high. So actually just subsetting the counts and divide the expression by the distribution of the counts, of the values of the counts. I think, I don't know, I might be wrong, but
maybe you can use the normalized count, Jonathan, and see the general parameter, the general pattern of these genes, for example, and check if they are around the mean, around, or too much high of the mean, or too much lower than mean. I don't know. It's just an idea.
you have to do by gene, by group, right?
First, like, separate these data sets that you have to go deep. And after you check this, but as Thiago told you, you have to spend more time on this to like establish a method to define this.
Yeah, one thing you can try to do actually is try to have a large quantity of clusters, like use a good resolution to get a lot of clusters, and based on that, try to extract, for example, the mean expression for each of these clusters, and try to correlate the mean expression between these clusters. Because if you try to correlate these in the clusters, maybe you can see the effect of the for example, the increasement or decreasement of the markers that you're interested for. So I think this could be another strategy for that. But like Thiago said, you need to spend a good time and a great amount of time just through knowing the data set and working on that, because this is not a easy task to do, like comparing the different expression levels for these markers that you're interested for.
Okay, thank you.
Guys, I have another meeting, so I'm going to stop recording, but if you want to continue the discussion, feel free. Okay, so thank you guys for